Day 6, I'm going to introduce some of the cellular communication means. And here it is the direct cellular communication source. So as you are aware that there are multiple ways of cellular communication. One is direct communication, another is indirect communication. Direct communication, which I will be discussing here, that is junctions, and another one is via signaling molecules. So here there are some junctions that is here called intercellular junctions. So cells can also communicate with each other by direct contacts, that is referred as intercellular junctions. So these junctions are of different types. In plant cells there are different source junctions, and in animal cells different junctions. So plasmodesmata are the junctions between plant cells, whereas animal cells have different kinds of junctions. For example, tight, gap, and adherens junctions. And then desmosomes. So basically in animal cells, there are two types of junctions, which I will be explaining. Now, in general, the long stretches of plasma membrane of neighboring plant cells cannot touch one another because they are separated by cell walls. So plasmodesmata are numerous channels that pass between the cell walls of adjacent plant cells, connecting their cytoplasm and enabling signal molecules and nutrients to be transported from cell to cell, as you can see here. And so first one here, pathways through the cytoplasm. And here this is plasmodesmata. Then cell wall, cytoplasm, vacuole, as you can see, and you are aware about vacuoles. And this is pathways, the red color, that is for communication, cellular communication, and it is also called intercellular communication. Okay. So next is how it is made up of. What are the, for example, tight junctions, cell A, cell B, and then tight junctions. The junctions are here. Extracellular space, groups of tight junction proteins, and plasma membrane. In human, it is well-defined junction proteins. So they have really specific roles, and they play crucial role in human diseases. Now, you can see this one is for gap junction. So here you can see desmosomes of cell 2, intermediate filaments of cell 1, then intermediate filaments of cell 2. Then here plasma membrane, desmosomes of cell 1. So this is green color, and desmosome of cell 2, yellow color, extracellular space. So this is gap junction. Then desmosomes, and then it comes gap junctions. Here it is cell membrane 1 and 2 for both the cells. Then you can see one is called closed, another is called open, and that is connection. So this is basically the channel between animal cells, and gap junction channel is formed here. Then extracellular space is formed here. Now, if you want to understand the junction types, then as I mentioned, there are two types of junctions. One is here where you can say tight junction and gap junction. Another is anchoring junction, where you can include adherens junction, desmosomes, and hemidesmosome. So in adherens tight junction, you have seen it. A gap junction, something it should look like gap between cells. And tight junction, here you can see adjacent plasma membranes, intermediate space, intercellular space, then strands of transmembrane protein. So then it comes anchoring junctions here, adjacent plasma membranes, like this one. Then similarly, you can see different structure. And then there are some differences between the compositions of the junctions also. So what you need to know is the components and their functions, junction functions, So which is here? Tight junction seals neighboring cells together in an epithelial sheet to prevent leakage of molecule between them. Adherens junction joins an actin bundle in one cell to a similar bundle in a neighboring cell. Then desmosomes joins the intermediate filaments in one cell to those in a neighbor. Gap junction allows the passage of small water soluble ions and molecules. Then hemidesmosomes anchors intermediate filaments in a cell to the basal lamina. Now you can see these junctions with their components in a separate figure. For example, tight junctions, occludins, claudine protein, junctional adenosine molecules, then adherens junction, cadherin, adhesion receptors, cytoplasmic proteins, gap junction, connexin, transmembrane proteins, desmosomes, cadherin, family proteins, desmoglein, and desmocholine. These are more than enough up to for up to +2 level to understand, and you can make hundreds of questions from here. So try to understand these figures and their functions and their components only. Now, a tight junction is a watertight seal between two adjacent cells where protein holds the cells tightly against each other. As it appears from the name, this tight junction adhesion prevents materials from leakage. Now, these tight junctions are typically found in the epithelial tissue. So these are some objective type of questions, multiple type questions where you can make it. And then tight junctions, after tight junctions, you can go in also found only in animal cells. So this is another question answer. That is desmosomes, which act like a spot welds between adjacent epithelial cells. They keep cells together in a sheet-like formation in organs and tissues that stretch like the skin, heart, and muscles. Then gap junctions in animal cells are like plasmodesmata in plant cells. So this is another question which you need to understand between plant, what is the plant cell and what are the similar junction in animal cell. So the junction for the plant cells and the junction for the animal cell. Here gap junction similar to plasmodesmata. Now there is another repeated thing which I am presented, the components of prokaryotic and eukaryotic cells and their functions, which I have presented several times in several short and long videos. So you need to have a kind of map in your mind. For example, flagella, function, cellular locomotion, and then present in plant or animal cells, prokaryotes or eukaryotes, where it is present. Then cilia, cellular locomotion, movement of particles along extracellular surface of plasma membrane, and filtration. So these are some of the things. Mitochondria, what is their function? Peroxisomes, their functions. Cell wall function, Golgi apparatus function. So this is summarized view for all these cell components, which you need to know, whether you are in class 10 or 11 or 12. So you need to know the components of prokaryotic and eukaryotic cells and their respective functions. The same thing, you can also see here some of the differences in the form of Venn diagram between prokaryotes and eukaryotic cells. Now you can go for cell membrane, which is quite easy, but you need to remember few things. For example, what is fluid mosaic model, and then what are the compositions of cell membrane. For example, if you remember something which I have discussed was like glycoprotein, what is, for example, DPP, and then the water, where the water is located, what are the proteins which are there and why there are some protein channels, what are these cholesterol doing here. So all the components of cell membrane you need to know. And if you want to know the real composition in summarized way, you just need to know protein, cholesterol, phospholipid, that is what I was saying, DPPC, and then polysaccharide attached to protein forming a glycoprotein. And then there are some protein channels. So from the name itself, you can understand what is the function of protein channels. So first you need to understand this structure, which is simple form of the fluid mosaic model for plasma membrane. If you understand this, then go to this part, and then you can understand the real perspective of cell membrane and their compositions. And after understanding this, you need to understand the function and main properties of cell membrane. So in terms of properties, you can say it acts like a barrier, controls the movement, it acts for the cell signaling and cellular recognition. So these are the summarized presentation for the property of cell membrane, and you can see how a cell and the cell membrane could look like, what are their components, and how the phospholipid bilayer looks like. So if you have a cell and the cell membrane, you want to understand, it could look like this, and the phospholipid could look like this. There are different types of phospholipids which you can see, and that is like with one of them is the PPC lipid bilayer. So next part is coming plasma membrane structure and function. The plasma membrane separates the internal environment of the cell from its surroundings. This is first and clear view of the function of cell membrane. Then it acts, so this membrane is a phospholipid bilayer with embedded proteins. Then plasma membrane has a fluid consistency and a mosaic pattern of embedded proteins. That is why it is called like fluid mosaic model. Then if you want to understand the major functions of the cell membrane, that is selective permeability, active transport, cellular communication, and cell adhesion and recognition. Then if you want to understand the summary of this plasma membrane, it is like the membrane is the cell's flexible outer limiting barrier that separates the cell's internal environment. It is present in both prokaryotes and eukaryotes. Main functions include regulation of exchange with environment, that is selective permeability, and then sensitivity to the environment. Then it comes energy transfer and chemical reaction, and then a structural support, of course. So basically, the same thing could be summarized here: selective permeability, active transport, cellular communication, cell adhesion and recognition, or cell identification. Then it comes, this is the last part for today's. This is just one kind of relevant question. How, as far the topics, subtopics which we have learned so far from this today's class. So how viruses could infect a specific organs. First thing you need to know the viral infection and the organs where it could infect. So a specific glycoprotein molecules exposed to the cell surface of the cell membranes of host cells are exploited by many viruses to infect a specific organs. And these viruses are able to invade the cells because the cells have binding sites. Definitely on the cellular surface that viruses have exploited with equally a specific glycoproteins in their coats. So the cell is tricked by the mimicry of viral coat molecules, and the virus is able to enter the cells. Other recognition sites on the virus surface interact with the human immune system, which could be damaged, prompting the body to produce antibodies. Antibodies are made in response to the antigens, and these same sites serve as the places for antibodies to attach with it and either destroy or inhibit the activity of the virus. Unfortunately, these sites on some viruses, for example, HIV, are encoded by genes that exchange quickly, making the production of effective vaccine against the virus very difficult. So there are organs, for example, it could be lungs, liver, brain. There are many kidney where you can see or you might have heard that the viral infection could take place, and of course eyes. Now next part will be in the next classes where we will be discussing some kind of transport, active transport, passive transport. Thank you very much.